Our next speaker is Itai Sharon, and he's a postdoc from the Jill Banfield lab. And if you've been following the research of Jill Banfield, she's really been focusing on microbial community structure and function in natural systems. And the method she's been focusing on is community proteomics, as well as metagenomics. And Itai today is going to talk about a technology that's called molecular, which is a long-read sequencing technology. And he will use that, I hope. I'm looking forward to see some data on how he's going to use that to look at strain variation and also get genomic information from organisms that are low abundance in ecosystems.
Okay, I would like to begin by thanking the organizers for giving me the opportunity to present my work. Hope that everybody can hear me. Okay. So today I'm going to tell you about our experience with applying the molecular technology for the study of microbial communities in groundwater sediment. Okay, so I'll begin with a short overview of the technology. So Molecular was a startup company that developed a technology that enables the sequencing of long reads that spans over thousands of base pairs. So the company was bought by Illumina about a year ago, and I'll give you just a short description of how the technology works. So the basic idea is to take the DNA, shear it into 8 kb molecules, and then amplify each of the molecules, and then shear it further so that it can be sequenced on HiSeq machine, and add barcode, and then do the sequencing on the HiSeq machine. And after that, all you have to do is just collect the reads based on the barcode, where each group of reads that comes from the same molecule will have the same barcode. The next step is just doing an assembly for these sequences, which is pretty easy because these are like 8 kb pieces, so it's not too difficult. So we use this technology for the study of microbial communities in groundwater sediment, and this is in a site called Rifle. And in this specific experiment, we were interested in what we call background sediment, and these are microbial communities that live in a place where no amendment experiments were done previously. We and other labs are doing this type of experiments in this site, and we were interested in just getting a sense of what is the natural community of microbes look like in this environment. So in order to do this, we collected 3 samples from 3 different depths. We extracted DNA and then did sequencing with both Illumina HiSeq and the molecular platform. So for the HiSeq platform, we got reads that are 100 base pair long, and from 4 lanes of HiSeq, we got between 34 to 74 gigs per sample. So for the molecular data, we got 3 libraries for each of the sample, and on average, the amount of data we got is about half a gig, with a read size of the average read size of about 8 kb. So this is just to give you an idea of what read size distribution looks like. So basically, we get read sizes that are between 1.5 kb up to almost 20 kb in some cases, where the average, as I mentioned, is 8 kb. Okay, so previously, before we got the molecular data, we assembled the Illumina data and got several near-complete genomes and also 1 complete genome. And this is the genome of RBG1, which is the first representative of a new candidate phylum called Zixibacteria. And the RBG1 genome is 2.2 megabases, and we were able to reconstruct it, the whole genome, from the short-read data. So if you look just at the short-read data, then RBG1 seems to be the most abundant genome in the community, as you can see here in this rank abundance curve. And just keep this in mind because you'll see that things get seem to be a little different when we also add the molecular data. So one thing that we wanted to do is to confirm the assembly of the RBG1 genome. And in order to do this, we identified reads, molecular reads that belong to RBG1 and compared them to the assembly of RBG1 genome. And as you can see, we found 2 local misassemblies. Neither are chimeric, but the majority of the reads, like 90% of the reads, matched perfectly the assembled genome. And the remaining reads had like small differences that are probably due to strain variation. So I think that in many cases, people question the quality of genomes that are reconstructed from metagenomic data, and I think that this show, despite the fact that we had 2 misassemblies we were disappointed with, then I still think that it shows that in general, once the proper quality control and manual curation procedures are kept, I think it's that high quality genomes can be reconstructed from metagenomic data. So when the contact was first made with Molecular, we were very happy about this because we knew that these samples are very interesting with a lot of exciting genomes, and we were hoping that with the molecular data, we will be able to close many of the genomes that we assembled using the short-read data. But as it turned out, this was not the case, and the reason was that the community was much more complex than we expected. So if you look in this figure, for example, you can see the coverage. We calculated the coverage by mapping the short reads to the molecular data and the Illumina assemblies. And as you can see, the majority of the molecular reads, this is for the 5-meter sample, the majority of molecular reads have 0 or 1x coverage, which means that they basically come from the long tail of rare genomes in this environment. And because of this, we were able to do only a small amount of assembly using an assembly program that we wrote, and also do some scaffolding, but not as much as we expected. So I think that this is something to keep in mind, that either we have to have more molecular data, or I think that if this technology is applied to less complex communities, then it will probably be possible to get more scaffolding and assembly. So we wanted to know whether we see the same things with the molecular data and the Illumina assembly, and one of the things we did is to generate this rank abundance curve that is generated based on the gene that encodes the ribosomal protein S3. And in this case, every column represents a species where the color of the column tells you whether the RPS3 gene was found on the Illumina assembly, in this case in orange, or on the molecular reads in blue, or in some cases we were able to find the same genes, the RPS3 genes, on both types of data. And so we got much more RPS3 genes from the Illumina data, but then the interesting thing was that for the least abundant and the most abundant species, we found the RPS3 genes mostly on the molecular data. So if you think about the least abundant organism, this pretty much makes sense considering the previous slide I showed you. So many of the molecular reads come from rare genomes, and therefore it's not surprising, and since they are long enough to recognize genes on them, then it's not surprising that we are getting many RPS3 genes from these reads. But then we were quite surprised by the fact that we found that some of the most abundant species are represented only on the molecular reads. And in fact, if you look at the RBG1 genome, which from the short read data seemed to be the most abundant genome, then you can see that here it's only the second most abundant genome. And when we checked it further, it turned out that the reason that we didn't see much of these species in the short read data is because of strain and species variation. So 5 of the 7 most abundant species in this case are closely related species from the same Delta proteobacteria lineage. And as some of you may know, short read assemblers tend to fail with the presence of strain and species variation. So we were interested in getting to know more about these most abundant species that we found. This is in the 5-meter sample. And in order to do this, we clustered the molecular reads based on overlap, so 90% identity overlap, and we were able to generate 4 clusters that had at least 100 reads in them. And then we also did gene prediction, and we clustered the genes into gene families. And when you look at the phylogenetic profile of these 4 clusters that you can see here, then it seems to be pretty similar, with the largest piece of the pie coming from Delta proteobacteria. So one of these clusters was significantly larger than the rest. This is cluster 1 that you can see here. One other thing that we did was to try and reconstruct genome architecture for these species and strains, at least for the region on these species and strains that is common to all of them. And in order to do that, we developed an algorithm that, by considering the molecular reads and the order of genes on them and the gene families, reconstruct synthetic regions for the genomes. So we were able to reconstruct the genome architecture for the majority of the Delta proteobacteria clade of strains from the 5-meter sample, and also for another clade of strains from the 4-meter sample. In this case, these are OP8 strains. And we were able to reconstruct regions that are more than 100 kilobase long. So I would like to finish with this slide, where in this slide you see multiple sequence alignment of molecular reads that all fall on a conserved region on the Delta Proteobacterium genome. This is the region that encodes the ribosomal proteins. And as you can see, I hope, then in the molecular reads it is possible to see that there are quite a few SNPs. And in fact, when you play with this a little bit, then I was able to reconstruct 9 different strains from these molecular reads. And then when you also consider the Illumina data, then you can find even more strains. And I think it's pretty fascinating to see that we have this one lineage that is represented by so many species and strains. But then when you consider RBG1, then for RBG1 we didn't find any SNPs on this region and only a few mismatches. So I think it's something that is very interesting to understand why certain lineages are represented by only a small number of strains, while others are represented by so many. So, okay, with that I would like to finish. I would like to thank people from the Banfield lab who helped me on this project, and also people from JGI and Molecular. And thank you.
Great. We have time for 1 to 2 quick questions before the break. Any questions? I have a very practical question. What are the DNA requirements for the molecular?
I'm not sure.
Okay. And I guess one other question. You had the RGB1 genome, and you were showing the differences between the molecular versus your reference. And you said there were 2 misassemblies, but then there were also other differences. How did you define the other differences versus the misassemblies?
Okay, so the way we did it was by in every place. Sorry. Okay. So in every place where we found differences between the molecular reads and the RBG1 genome, we went back to the short read data and we mapped the reads to the genome and checked whether there are misassemblies there or whether the short read data is consistent with the genome. So whenever the short read data was consistent with the genome, then we concluded that this is either something that had to do with strain variation or maybe something else.
Okay. We have one more question in the back.
Oh.
So the question was what percentage of the reads we were able to assemble? This is what?
You should be able to tell the ends, right? So then, how many? I mean, roughly, is it 50% of the molecular read that can go from one end to the other end, or it's only 10%?
So I'm not sure, like, what ends are you referring to?
When the molecule they're amplified, they start from roughly AKB region.
Okay.
They get amplified. When they amplify that AKB region, you have a distinct end because there's a primer on the end, so you know these features. So my question is just that when you assemble the fragment from the Illumina, what percentage of the molecule can you see the end?
So I don't know exactly because we didn't do it like the molecular people did it, but I think that from the data we have, I think that the majority, probably 99% of the molecules, something like that.
I think there was one more quick question, Manuel.
Yeah, I was wondering, could you use that to sequence complete 16S genes for tag sequencing, basically, that you would get a whole gene?
Yes, so the answer is definitely yes. So for example, for the Delta Proteobacteria clade from this 5-meter sample, we were able to identify 14 16S genes, which was much more than any other species in this, or any other OTU in this data set. And with respect to percent identity, for example, average percent identity was higher than 99%, which also support their being part of the same clade, like close species of strains. I couldn't hear you.
Could you use the PCR product, the 16S PCR product? that into the molecular process and essentially get
Oh,
these out.
I assume yes. I
It
mean,
would probably
yeah.
be an expensive way to do
Yeah.
that, but
Yeah,
maybe
it's possible.
stagger sequencing.